Hello, my name is Daniel Olson, and I am a data standards developer and a member of the data science team at Critical Path Institute. Thank you for joining me today to talk about data standards and integration. Why do we care about integrating information? According to CDISC, the Clinical Data Interchange Standards Consortium, the true measure of data is the impact it has, but without integration, its full potential is not realized. So the purpose is simple. It's to reach that full potential. It's to amplify the impact of data. And I think this is especially true for rare diseases because a single study is not going to be able to capture all of the disease progression and all of the important and critical information about a given disease area. But by integrating multiple studies or registries or other type of relevant data, we can provide additional statistical power to find meaningful differences in the data and help generate new hypotheses. Now, without going into probability theory, in general, when comparing groups with frequentist statistics, which are very common in clinical trials these days, given the same magnitude of effect, if you have more subjects, then you have more statistical power to find those significant differences, those things that are going to support your hypothesis. Furthermore, by integrating information together, that diversity can lead to new discoveries to help us generate new hypotheses about a given disease area. One example of that is c-path is launching this new consortium called CureID. It's a public-private partnership set to provide a forum for the exchange of clinical practice data to inform potential new uses of existing drugs for areas of high unmet medical need, including COVID-19. So by combining clinical trial information that may have similar phenotypic outcomes to COVID-19 or to other diseases, you can take information that exists and repurpose that information for something new, to generate a new hypothesis to say we've used this drug for these patients with a similar phenotypic issue, and we've seen these results. Perhaps that would also work with somebody with COVID-19. So if it's so important to aggregate data and it provides these benefits, why isn't it done more frequently? And I think really it's mostly because it's a messy, difficult process. Everybody has their own bespoke way of collecting information, referring to variables, values, procedures, and each study has its own endpoints. So they're not collected in the same way. They don't have similar purposes in how they're collecting the information, and they have different systems to capture that information. And so pulling it all together is not it's not as straightforward. So one of the organizations that's working towards a systematic analysis of disparate observational information is OMOP, and they have a common data model. And I like this information from them because it sort of gives a nice way of representing what we mean when we mean messy data. And so in this infographic on the top right, we have the raw data represented as outlets from various parts of the world. And so if you have a two-prong plug from North America, it's not going to work everywhere without some type of adapter. So if you're traveling and you forget your adapter, you're going to be in bad shape. However, they've sort of used a verb here where they've OMOPed the data, and it's been transformed into a standard so that it's the same for everyone. Now, I'm an American, but I live in Japan, so I especially like this graphic because Japan only uses the two-prong approach here, and so if this OMOP data would make my life a lot easier. So if we could just get everybody on board to in the world to make these plugs, then that would be great. It would also be beneficial for for manufacturers. Imagine having to create a different plug for a device that goes around the world and goes into every one of these different outlets. It would really simplify things. So the second graphic below that sort of shows the structural differences in how source data has been collected, how it can be transformed using the common data model, and the output of that is information that is in a uniform fashion so that it can later be analyzed. So, as we showed in the previous slide, standardization is sort of the way forward for integration. So it's hard to integrate messy data. If you standardize it, it makes this integration process a whole lot simpler. And one of the organizations that has dealt with messy data for decades is the FDA. So the way they describe data standardization is that it provides a consistent general framework for organizing study data, including templates for data sets, standard names for variables, to helps identify appropriate control terminology, and shows standard ways of doing calculations with common variables. So for them, it really is helps them review process and receive review process and archive submissions more efficiently and effectively. So much so that they've decided to require them for all new submissions. So both FDA and PMDA here in Japan require CDISC standards for drug submission because of how much it affects their review process. But for us, I work with Critical Path, it also helps us to have things in a standard way so that they can then be integrated. So there's two sort of main overarching types of standardization I want to discuss briefly. One is semantic standardization, the other is structural standardization. Now, semantic standardization really just means using controlled terminology to say the same things when we mean the same things. Now, I like this. Nathan Pyle is one of my favorite comics, and these aliens understand each other, and we can understand what they're saying, but data sets are not as simple as they're more simple, so they can't really process natural language in the same way that we can. If I said I chopped my own flavor muscle, you'd know that I meant I bit my tongue. But within a data set, it's a lot easier to just say the same thing when you mean the same thing. And there's an example on the lower left. So if we wanted to combine 3 studies using a scoring, the left side you can see the score. It wouldn't be possible with these 3 studies because of the way that they have tagged their adverse events. The information for mild, for example, is broken out into very mild and mild in study C. So we could technically combine those two, but it may not be possible. There may be enough difference there that to say that mild and very mild are systematically different than mild in the other 2 studies. So we can't combine that information. And furthermore, if we just wanted to combine the terms and not combine them in a score, we would run into issues where study A has, for missing data, they have absent, not assessed, or unknown. Study B uses not assessed or missing and concatenated into one field. And study C uses missing or unknown concatenated into one field. And those are have different meaning. If something is not assessed, that's a known. We know that it was not assessed. So study A has broken it out into 3 different terms, and so combining those with study B and study C just wouldn't be possible, because we don't know if missing or unknown in study C means the same thing as not assessed in study A. So we would run into issues where the semantics are important enough that we need to know what happened to this missing data, and combining them would be problematic. So the semantic standardization, CDISC calls it control terminology. This is taken from one of their webinars. I just wanted to sort of show that it's so important that they say that it underpins every effort they have in standardization. And there's a lot of standardization that takes place at CDISC. They have protocol standards, the PRM. They have data acquisition standards, C-, which is the clinical data acquisition standards harmonization. All of that information sort of is for annotating CRFs that will basically allow information to go directly into SDTM, which is the study data tabulation model, and that has traceability to the final analysis data and results, which is just the ADAM model. But the control terminology underpins all of this. And so it's such an important topic that there are many sort of reference terminologies out there for different sort of specific needs. For example, rare disorders, Orphodata has its own ontology that sort of has that gives you a hierarchy of terms in a systematic manner, but it cross-references like the ICD-10 for diagnosis and MEDRA for like adverse events, that type of information. So using one of these vetted, validated reference terminologies whenever and wherever possible is critical, because without doing so, it could lead to sort of data loss along the way. And it also provides the additional metadata so that even if you're using different reference terminologies, by looking at those reference terminologies with the supporting information, you can sort of get a good idea of whether those will be compatible and can be standardized into yet something else if you need to. If you want to combine them, you'll have all the supporting information to combine those terms. Whereas if you just come up with terms on your own, you sort of take a gamble of whether that information will be compatible later with anything else, or whether it's just going to be useful for your one study. So the second type of organization or standardization is structural standardization. This is an example of a CDISC exposure domain. They've grouped the identifier variables on the left, the topic variable, and all the qualifiers in the middle, the timing variables to the right. So the variable order is specified. Every variable name is specified and can be referenced within a study data implementation guide so that everyone knows, everybody can refer to the same documents and know what they're talking about when they use those variables. There's no, it's not ambiguous at all. And they also specify the record structure. So for a lab domain, they permit one record per lab test, per time point, per visit, per subject. So that tells you the structure of each row. Each row must be unique per lab test, per time point, per visit, per subject. So if we had just had a structure of one record per subject, then that would completely alter the organization of that database. It would be a very wide database where someone had a full clinical chemistry panel and you had to represent those each in its own unique variable. It would be very wide, whereas the example structure shown here would be very long. So just a brief recap. Data integration is important because it amplifies the impact of data that's been collected. Data standardization can support that integration. And there are two sort of main overarching types of standardization, semantic and structural. So I just wanted to briefly recap before we kind of talk about the actual steps to integration. And I'm going to use critical path as an example. So critical path, we have to go through, we need lawyers involved to create data use agreements, data contribution agreements that need to be signed by both the data contributor and critical path. That's not something we can get around. It sort of protects everyone. Once we have those source data sets, we review all of the documentation related to those protocols, data dictionaries, the manual of operations, all the associated metadata. We explore them so that we can better understand the topic elements, the concepts, the variables included, sort of what went into the study itself and how the information was collected. Once we have a good understanding of that information, we compare that information to a standard. And now this is commonly a CDISC standard, along with reference terminologies like MEDRA or WHO-DRUG, or NCI EVS has Codelists as well. And we identify the gaps to the standard. Now, sometimes everything lines up and we have all the information we need. It's just in a different structure, but it lines up. Sometimes there are gaps within the standard where there's some novel biomedical concept that just hasn't been included in a CDISC standard or model, or there's some terminology that just hasn't been adapted into a controlled terminology list that we can reference consistently yet. So we have to take the time to develop strategies to address those gaps, and that can be a time-consuming and difficult process. Once those have been addressed, we create this mapping specification, which is like a roadmap that is developed to basically say, how is this source information going to be transformed into this standardized target information? Then we write programs in SAS or R to transform that information, and then we QC it, and then finally we can combine all of that information. So this process takes a long time, and there are places in here where if data was standardized from the outset, we would be able to reduce the amount of effort it takes. And unfortunately, when folks don't use a standard during this exploration and gap identification phase, sometimes we discovered that the information has been collected in a way that's just not possible, or it's just not prudent to combine them. For example, that adverse event severity would have been a problematic set of information to combine. And I've actually seen information where we've been given lab data. It's from a legacy data set, and it had all kinds of supporting information, and it looked great, except that it was missing one critical element, which was the units. So there was no way to tell whether the clinical chemistry or, the information that was collected for like glucose was in milligrams per deciliter or nanomoles per liter, or how it was collected. So that data, even though the sponsors thought it was collected and put together in a way that made sense to them, because maybe they only have one way of doing glucose and they only use milligrams per deciliter, so they just chose not to include that because they know what they're talking about. When that information came to us, without going back to the source information, sometimes the information is 10 or 15 years old, you have no way of knowing what units were used. So that just leads to irretrievable data loss. It just has to be thrown out. And so we hate to do that. And I think this is also true for registries. because registries, it's not mandated by, let's say, the FDA to collect registry information in a very specific standardized way. And so registries can sometimes collect information in a way that makes sense to them. It's logical, it makes sense to their one unique study, but then that information, it can't be used to have a greater impact later because it can't be combined with other information because of the lack of standardization, so that, it's fine for their one very specific piece of information, but then you just can't combine it with other information and allow that data to be impactful to a greater audience. So here's some of the benefits of standardizing information. Really, the goal would be to just sort of automate this exploration phase and to automate the transformation phase by creating computer programs that just make sure that the information that's been sent is correct, and then pull that information together in a really simple and straightforward way because it comes from a standard. It also sort of reduces this middle part where we have to identify the gaps and fix these standard gaps and develop specifications for mapping the information and roadmaps and all of that. Because if it's already come in a standardized format, we already know what's in there. We don't have to sort of figure out whether someone meant that a 0 and a 1 in gender, the 0 was male or the 0 was female and the 1 was male or female. We don't have to go and figure all of that out because it's been collected in a standardized way. We already know what the information means, and there's no gaps to address there. So the hashtag goals for RDCA-DAP, the Rare Disease Cures Accelerator Data Analytics Platform, is to sort of create this collaboratory, this sort of repository that can be an analytics hub to promote sharing critical data across rare diseases and accelerate the understanding of disease progression. Which includes, clinical trials, longitudinal studies, natural history studies, any real-world data that we can use and combine for this. This is the payoff here. All this information on the left being funneled into the Data Collaboration Center, where it can be curated, standardized, aggregated, and integrated into something that can then be used for actionable rare disease drug development solutions so that we can generate those new hypotheses and have the power the statistical power necessary to find differences within and between groups. And Critical Path has done this before. We've already proven that this aggregating clinical trial information to develop new disease progression models and clinical trial optimization tools works. We have a track record with this, but now we have an exciting opportunity to do so in the rare disease space. I really appreciate your time and attention today. Thank you very much.